good afternoon. It's my pleasure to welcome you to the annual George Corey Lecture. This lecture series was established to remember George Corey, one of our most distinguished lab chiefs at the NIH, who died almost 25 years ago. The anniversary of his death will be next April. It was created in part by his very dedicated postdoctoral fellows and trainees, by his colleagues at the NIH, to remember our truly exceptional scientists who worked at the NIH for all too little time. I'd like to thank all of the people who've been working and putting together this program, but especially T. Zhang, who for years has worked pretty tirelessly to create what I think has been a spectacular lecture series that we can use to remember George. So who was George Corey? I see people in this audience who may not be much older than the anniversary that I just mentioned. He was a graduate of Princeton, was a Harvard Medical graduate, and in fact a classmate of mine at Harvard Medical School. He did his internship and residency in medicine at the Mass General Hospital and then came to the NIH, where he worked with Norm Saltzman, another very distinguished and legendary virologist. He conceived of the idea that SV40 would be a wonderful model in which to study both the development of cancer, it is a tumor virus, and also, because it was a very simple DNA virus, how viruses worked, how transcription was regulated, and so on. And he made what I would call really seminal discoveries in the area of transcription regulation, and in fact was probably the first person to describe enhancers in the SV40 system. The photo that you see here was taken in 1983 at one of the many events. You can recognize me in that picture, and that's Susan's nose, and at the bottom, Bruce Howard's head. And the other person here is Linda Paston. So we were eating or drinking something, which was fairly typical in those days. Nowadays, doesn't happen very much anymore. George really had a phenomenal career at the NIH. In addition to the wonderful science he did, he mentored an extraordinary number of speakers. And in fact, in this series, for the first 10, 12 years or so, we just really invited his postdoctoral fellows, and they were all really terrific speakers and have had outstanding scientific careers. He loved science. He loved to talk about science. He loved the people he worked with in science. He was enthusiastic but critical in a very positive kind of way. And he was an enormously generous person. He was always interested in the science you were doing and was very involved in the careers of many, many people. He helped his alma mater, Princeton. He helped the Wistar Institute, where another lecture series exists in his name. And he was just an enormously popular and very positive presence at the NIH and in the entire community. The year of his death, he was elected, not actually just a few weeks before his death, elected to the National Academy of Sciences at the age of 43. It's sobering thought that nowadays your average NIH grantee doesn't have a grant until they're 44 years old. And George already was an incredibly accomplished scientist. I think that reflects not only the times, but more importantly, his amazing talent as a scientist. One of the great pleasures that he had was going to scientific meetings. And I was rummaging around in my archives and came upon, let's see, this picture, which was taken in 1980 at the Animal Cells and Viruses Conference. And it may be a little hard with the lights on and the size of the picture to make out faces, but this particular picture is full of not only legendary NIH scientists, but legendary scientists who really created the field of molecular virology. And I think I have an enlargement so you could see that George was here. There he is. I was there. There are other people here, people like David Baltimore and Mike Bishop and Marilyn Kozak, and this is Norm Saltzman, with whom George worked, and on and on and on. I actually brought the picture with me if anyone is interested in seeing a slice of history. This is a wonderful example. In addition to loving his science, George was devoted to his children, David and Lisa, and his wife, Marilyn. Marilyn Corey is here in the audience, and I welcome her. She attends every one of these lectures, both at the NIH and at the Wistar Institute. And based on the number that have occurred since George's death, I think she deserves an honorary degree in molecular virology. She also listens very carefully. So on that note, I would like to introduce Kathy Zune, Scientific Director of NIAID, who will introduce this year's speaker.
Thank you, Michael. Before I introduce Diane, I just wanted to make sure people were aware there will be a reception after Diane's talk today, and it'll be in the library next door, so it should be convenient to everyone. Well, it's a great pleasure for me to introduce Diane Griffin today. And she's not only a colleague, a friend, but also a very important contributor to the NIAID's Board of Scientific Counselors, for which we are extremely grateful. So it's quite an honor for me to introduce Diane. Diane grew up in Oklahoma City, Oklahoma, where her dad was a geologist and taught Diane really the love of science and the love of teaching science. And she kept up the family tradition and attended Augustiana College in Rock Island, Illinois, where she earned her Bachelor of Arts. She went on to Stanford University, where she began her training for her PhD in microbiology. And there, her interest was piqued in medicine. And she applied for and was accepted to Stanford's MD PhD program. At the convincing of her mentors, it wasn't easy, but they encouraged her very strongly to go for both her MD and her PhD, which she did. And she actually wrote her PhD dissertation while she was on a month's vacation from her internship at Stanford Hospital. So it shows you the kind of perseverance Diane has. Following her internship and residency also at Stanford, she decided to focus on research rather than clinical medicine, which is all very good for us. And she moved with her husband, Jack, to Johns Hopkins, where she did a postdoc with Dick Johnson and really became interested in viral infections of the nervous system. She then started her studies on pathogenesis of Symbis virus, a murine alphavirus that causes encephalitis. And she later collaborated with Dick in studying the neurological effects of measles virus, particularly focusing on measles encephalitis. In 1973, she became an assistant professor at Johns Hopkins Neurology Department and ultimately became a full professor there in 1986. In 1994, she moved across the street to the Johns Hopkins Bloomberg School of Public Health, my alma mater, and we do a lot together for that, to become chair of the Molecular Microbiology and Immunology Department. 2 years later, Diane's research deciphering the mechanism underlying the profound suppression of cell-mediated immunity accompanying measles was published in Science. She found that measles virus could suppress the release of IL-12 from monocytes and thereby depress cell-mediated immunity. This helped to explain why children were more vulnerable to other infections after a bout of measles. Diane is currently the University Distinguished Service Professor and Alfred and Jill Summer Professor and chair of the W. Harry Feinstein Department of Molecular Microbiology and Immunology at Hopkins. She's an editor for Fields Virology and has published more than 300 papers. She's served on several editorial and advisory boards and is past president of the American Society of Microbiology, the American Society of Virology, and the Association of Microbiology and Immunology chairs. She is a fellow of the American Association for the Advancement of Science and a member of the National Academy of Sciences and the American Academy of Microbiology and the Institute of Medicine. So again, it's with great pleasure that I introduce Diane. Thank you very much for coming today, and we'll hear about viral clearance. It isn't easy.
Thanks. Well, it's a real pleasure to be here and to be able to give this special lecture. Unfortunately, we don't have a way to change slides up here, so I'm going to hear a lot of nexts here. So next. So I didn't know George Corey. I would have hoped that our paths had crossed somewhere along the way, but he was a DNA virologist, I'm an RNA virologist, and so I didn't know him. But when it was announced that I was giving this lecture, next, I got an email from a colleague who is Connie Trimble, and I thought I would just read a few lines from her email because she reminisced about, she was a next door neighbor to George, and he was an important mentor to her. When I was a kid, every summer, my parents would park me with one of their colleagues at an NIH lab. I spent 2 summers pouring sticky Sephiroth columns as big as I was, another summer pouring 200 agar petri dishes a day, and finally it occurred to my parents that I might work in our next door neighbor's lab. For 2 summers I rode in with George, and he would wave his hands around and dispense wisdom, some of which had to do with science. I babysat George's kids. I house sat for them when they were away. George would tell me it was okay to bring boys, but don't tell anyone. And I remember lumbering over there a couple of times and just having dinner with them myself. All wild stuff for a first-generation immigrant geek. He came up for house parties my senior year, and I assume this is at Princeton, and was my date. He was so funny, he decided he would pretend he was an old curmudgeon alum. We went up and down Prospect Street, and at the front door of a club, he would introduce himself as an alum of whatever club it was. No, I do not remember how he danced. I've blocked that out completely. I pooped out before he did. He was good about sticking up for me to my parents. George says it's just fine not to major in science. George says it's okay for me not to be a pre-med. Okay, I'll stop reminiscing. I've outlived George and haven't done half as much. So next, so I just comment that she's now working at, she's on the faculty at Hopkins and is working on human papillomaviruses, which was one of the viruses that George worked on. So I think his science also influenced her career decisions. Next. So I'm going to talk about virus clearance, and in the context primarily of RNA viruses, because they mainly cause acute infections. But just to put this in some perspective, these are the different sort of varieties of clearance and non-clearance models for a number of different virus infections. So next. So if you talk about persistent infections, these are viruses from which you can continue to recover virus for usually the lifetime of the infected individual. And we know lots of viruses that do this. They just don't get cleared, and infectious virus continues to be recoverable. So certainly retroviruses do this. HIV, hepatitis C virus, B virus, and human papillomavirus often is not cleared either. Next. And then there are viruses that have an acute phase, and then later, then a period of time during which you cannot recover virus, but where you can identify it by molecular techniques. And then later, you can recrudesce. And most of the herpes viruses are reactivated like that. And next. So most of the viruses, and the ones we're going to talk about today, are thought to fit into this category. Acute infections, you're sick, you live or you die, you clear the infection. After you've recovered, you're immune to that infection again, and to getting that infection again. And so we've been interested in how these clearance mechanisms work. So next. So the systems that we've studied, as Dr. Zun has mentioned in her introduction, have been they're both RNA viruses. They both replicate entirely in the cytoplasm, so they don't have a chance for integration into the genome, et cetera, like DNA viruses do. So it's an alphavirus that causes encephalomyelitis. It has a mouse model. And then secondly, measles, which is a human virus, so the best model we have is a monkey model. So next. So we're going to focus first on the alphaviruses next. So these are simple viruses that are mosquito-borne. They cause encephalitis, or they cause rash and arthritis in humans. They're plus-stranded RNA viruses that have good molecular approaches to studying them. So again, simple viruses, 4 non-structural proteins, and then a structural, 3 main structural proteins that are translated off of a subgenomic RNA. Next. So these 3 structural proteins are put together in the capsid, a single capsid gene that surrounds the RNA, and then the 2 surface glycoproteins, E1 and E2, which heterodimerize and then trimerize to form these flower-like structures on the surface. It's an envelope virus, so that's depicted in this green here, the envelope of the virus. Next. So Synbus virus, the model that we're studying of alphaviruses, are a particular interest to us because the target cells are neurons. And in the nervous system, it doesn't really infect other cells besides neurons. So here's a in situ hybridization of a spinal cord of a mouse that is infected, and these are anterior horn cells, the motor neurons in the spinal cord, which are particularly susceptible to infection. And there's lots of other cells here, but only the neurons are infected. And this also makes the point that all the replication, since this is in situ hybridization, all the replication is occurring in the cytoplasm of these cells. Next. So it was recognized when this virus was first isolated that there was an age-dependent susceptibility to fatal disease being caused by this virus. So if you infect very young animals, virus replicates to high levels. The mice die within 2 to 3 days after infection. But we're much more interested in this pattern, which you see in older animals, that you get virus replication, not to nearly as high titer as with the young animals, but and then virus is cleared. And by 7 or 8 days after infection, you can no longer recover infectious virus from these animals. Next. So we've looked now a little more closely. I think next, I think there's a, yeah. Now, and this is a period of time during which there's infectious virus, and that's what is depicted here. And we've looked at both the brain and spinal cord. So again, in these animals that have followed over a much longer period of time, you can see that again, all the infectious virus is gone within 7 or 8 days after infection. However, if you look at viral RNA in the nervous system, you get a very different picture about the clearance process. First of all, you still get a peak at the same time, and you get a diminution at the same time that you're clearing infectious virus. But RNA actually clears much more slowly. Next. And over this next almost 2-month period of time, you have a sort of a steady decrease in the amount of RNA. And then after that time, it's a plateau. And RNA continues to be easily detectable using RT-PCR type approaches, and for, well, this goes out for 6 months, but for the life of the animal. So basically, you have 2 different types of clearance that are going on, infectious virus and then viral RNA, and then a period of maintenance of sort of steady state levels of RNA, or control of the recrudescence of that virus infection. Next. So some of the early studies we did to try to understand clearance were really focused on, well, before we understood much about the RNA situation, and we're focused on infectious virus and clearance of infectious virus. And so these are skid mice. These are mice that have no immune response, basically, no adaptive immune response. So they have neither T-cell responses nor antibody responses. And when you infect them and don't treat them with anything, they have persistent infection. So this virus doesn't kill the neurons, and it doesn't kill the neurons that it's infected. Doesn't kill the mice, at least not over this period of time that they're followed, but you continue, but the infectious virus is not cleared and not controlled. I mean, it's actually controlled, but it's not cleared. And so these studies use pretty simple passive transfer approaches to trying to understand what you needed to give back to these skid mice to make them actually able to clear the virus. And in these experiments, when we transferred T cells, we really had no effect on virus replication. But when we transferred antibody, and it started out this was just immune serum, but subsequently monoclonal antibodies, et cetera, we found that the virus actually was cleared very promptly. Within 48 hours, often we could no longer recover infectious virus. And it wasn't that it was just infectious virus, but also there was a clearance of RNA that paralleled, offset, but paralleled that clearance. But again, we could see persistent RNA after that period of time. Next. So, as I mentioned, we did a lot of monoclonal antibodies to try to look at the specificity of the antibody that was accomplishing this clearance mechanism. And this is a cryo-EM of Synbus virus, actually, with the FAB fragments of our favorite antibody that worked the best. And it was directed against the E2 glycoprotein. So a surface glycoprotein against one of basically the spikes on this protein. So it was against a surface protein, not against a replicase protein or something, and it was controlling virus replication. Next. Another experiment that colored our thinking a lot about how this antibody could be working was the development of, now we have 2 or 3 in vitro systems where this works, but the earliest one we used were dorsal root ganglion cells. These dorsal root ganglion cells, if you treat them with antibody, and this is the period of antibody treatment just for a few hours, after you establish infection, the cells survive, you treat with antibody, then you watch what happens to the virus. And when you could see if you use IgG or FAB'2 fragments, then the virus clears over a period of days. However, if you look at FAB fragments, there's really no effect on virus replication. But if you cross-link your FAB fragments with an antibody to FAB, now you do get virus clearance. So that told us that we thought that it acted at the surface and that they probably involved some sort of signaling that required this more than one binding site, basically. Next. So although you can think of a lot of other ways that antibodies might be effective against the intracellular virus replication, the one that we favor for a number of reasons. First of all, the neurons are not damaged by this process. Whatever it is that's clearing the virus is not hurting the neurons. In fact, they're doing better. So we think that there's a signaling process that affects virus, some part of the pathway of virus replication that then allows the cell to be able to control this virus, although not be able to completely get rid of the viral RNA, because as you remember, we continue to find viral RNA. Next. So now you need to know just a speck more about alphavirus replication to understand some of the studies that we did to try to understand how the antibody was working. First, early in infection, the cell is normal. It has normal cation concentrations intracellularly versus extracellularly. Has a good membrane potential with an active sodium potassium ATPase maintaining that membrane potential as well as the intracellular cation concentration. And the first thing that happens with any plus-strand RNA virus is you get translation of the non-structural proteins that then can start replicating the RNA. And then you eventually make this subgenomic RNA, and then you'll get translation of the proteins, the structural proteins that are going to go to make the virions that are going to bud from the surface of the cell. Well, during this process, in this later stage of the virus replication cycle, membrane potential is lost, sodium potassium ATPase function is lost, non-structural proteins are no longer made, host cell proteins are no longer made, so there's really a dramatic change in the cell that's ongoing as a part of the normal way this virus takes over a cell it infects. Next. So we looked to see what happened if you treated the cells with antibody. And this is just a dose response curve of different dilutions of antibody onto infected cells. And this is potassium flux, which is a measure for the sodium potassium ATPase function. And you can see that steadily improves with antibody treatment, and it's a dose-dependent improvement. Also, we looked at protein synthesis since the virus shuts down host cell protein synthesis. That also is steadily improved with the treatment, with antibody treatment. So next. So what it looks like is that, next, is that antibody has basically converted the cell that was looking like this at the time you treat it back to a cell that is that has more normal function. It's restored much of its function, both with respect to membrane potential, which actually this loss of membrane potential is thought to be very important for the ability of the virus to bud, so will inhibit budding. And then the ability for the cell now to make its own host proteins will also allow it to do things like make interferon responsive genes, etc. So you can imagine a number of things that could be improved by restoring this function. Next. So one of the questions then, we don't have complete answers, as you may guess, to how exactly antibody is working. It's still a very active area that we're working on. But one of the questions that all this raised was, what about T cells? I mean, T cells are supposed to be really important for virus clearance. And we'd shown antibody was important, but we hadn't necessarily really ruled out that T cells weren't contributing in some way or couldn't contribute. So PhD student Gwen Binder used B-cell deficient mice with the idea that if the animals can't make any antibody, then if there's virus clearance, then that must be happening due to at least some other aspect of the immune response than antibody, so a non-antibody mediated mechanism for clearance. Next. So we first looked at the brain, and we found that virus clearance in these antibody knockout mice at first looked like it was going to be pretty good, but then it settled back to being pretty much at the level of the skid mice. So in the brain, the animals were not able to clear virus. Here's wild type. But in the spinal cord, next, they were, we found a very different picture. First of all, the B-cell deficient mice were actually almost as good as the wild type mice. So this told us several things. First of all, T cells probably could clear virus from at least some populations of neurons. Populations of neurons that are mainly infected in the spinal cord are those that I showed you before, anterior horn cells, so motor neurons. And and it also said that neurons are different. They're not all responding in the same way because many different types of neurons are infected by this virus. Next. But it was also a question of how were presumably T cells working. We did many experiments that I'm not going to show you, but looked at and demonstrated that basically both CD4 cells and CD8 cells could participate in this process. Again, it was a non-cytolytic process. The neurons survived the clearance process. And so it seemed most rational that it was probably a cytokine that could be produced by both CD4 and CD8 T cells. So one of the other advantages to this handy-dandy alphavirus system, is that this subgenomic promoter that encodes for a gene, the subgenomic RNA for the structural proteins, the virus will tolerate putting in a second one of those subgenomic promoters, so you can make a second messenger RNA that's for whatever your gene of interest is. And so our gene of interest were a variety of cytokines, and we started off with the two that were at least known to have antiviral effects, and that was TNF alpha and interferon gamma, and then these are both controls. So if we look at the spinal cord, because that's where clearance occurred, you can see that this green here is the recombinant virus that is producing interferon gamma. And it is able to act essentially, convert these mice to very similar to the B-cell knockout mice with a control gene. So interferon gamma expression alone was able to clear virus from the spinal cord. So the second component to clearance was definitely interferon gamma, most likely from T cells. Next. So again, in vitro experiments to try to understand better how interferon gamma is working, and unlike the antibodies situation, this is a very well studied pathway in the immunology literature. Neurons have actually abundant interferon gamma receptors on them. And this again is an in vitro study looking at different doses of interferon gamma. These are the cells that are not treated, and so the amount of virus that's being replicated. And the suppression after treatment of virus replication in these cells by interferon gamma. Next. So, as I say, this is a well-studied pathway, and the best documented mechanism for signaling from the interferon gamma receptor is through the JAK-STAT pathway. And this looks at those same cells now. They're infected for 24 hours prior to being treated with interferon gamma. So we're really only interested in mechanisms that work after virus infection has been established in the cells of interest. So what can you do after the cell's already infected to get rid of the virus, basically? Infection itself, all these eyes at different times, are cells that are infected looking for phosphorylated STAT1, which is what you would anticipate would happen with interferon gamma signaling. And there's nothing, or actually interferon alpha signaling, but there's none that is occurring just with infection. But as soon as you treat with interferon gamma, you get a very robust phosphorylation of STAT1. And furthermore, it is very, very prolonged rather than being rapidly downregulated, which often happens in cells. So a prolonged activation of STAT in these cells. Next. And furthermore, Furthermore, if you block JAK-STAT signaling, there are some good inhibitors. This is what happens. This is the amount of virus being produced by cells that are not treated. This is the amount of virus that's being produced by cells that have been treated with the interferon gamma. And this is the amount of virus being produced by cells that have treated with a JAK1 inhibitor in addition to interferon gamma, so preventing that JAK phosphorylation. And you can see that that reverses this effect on virus replication. So the ability to decrease virus replication is dependent on that pathway. We still have yet to figure out exactly which proteins are of the 95 or so that are induced by interferon gamma in these cells that is responsible, and it's very likely that it's a mixture of factors. Next. So we also we're interested in getting back to the animal system a little more than the in vitro system, although in vitro system was definitely necessary. And looking at mice, and again, these are brains of mice that have different knockouts, so are deficient in different things. And you've already seen what happens if you're a skid mouse, which is deficient basically in everything, but my pointers died. So the top green line are the skid mice that are deficient in all adaptive immune responses. They don't clear. And if you knock out both gamma interferon as well as antibody, which is the second blue line, you find it's almost as bad. No the virus is never cleared. And the mu-MT mice, you've seen the B-cell mice, the GKO, each of those are sort of intermediate. The knockouts of the interferon gamma, or the GKO mice in the red, are, and see if this has come back, yes, they're interesting because they clear, but then the virus reactivates for a period of time before it's cleared again. So there's probably whole other processes that are necessary for maintenance of clearance, initial clearance, and then maintenance of clearance. Next. So now again, I think next, yeah. A recent graduate student has done a very detailed study of what's actually happening in the nervous system of normal mice. Now, to try to figure out who's doing what and when during this virus clearance phase. A lot of these other studies, as you can tell, have been done in immunodeficient mice of one variety or another, or in vitro systems. So this is a quantitating the cells that are coming into the brain after infection. So the green line are CD8 T cells. So the earliest cells to come in and the most abundant cells are CD8 T cells. This little box here is the first 8 days. So I'll remind you, during this period of time, all infectious virus is cleared. By the end of the box, you could no longer find infectious virus. So the cells that are there are, first of all, these CD8 T cells. The cells that are coming in next are CD4 T cells. And then the cells that come in somewhat later are the B cells. And so it's, we're in the process of figuring this out, but it's highly likely that it's a combination of these cells, and probably both the interferon and the antibody that's being produced locally that will be able to affect that clearance of infectious virus. Next. However, now, next. Yeah. So now if we look, so this is a period that we've just looked at. But now if we look out longer, and you'll remember now this is a period of time during which viral RNA is being cleared and then being suppressed or held at a low level. You find that these different proportions of these cells change over time, so that CD8 T cells actually diminish most rapidly. It's not too surprising. CD4 cells are maintained better. And then B cells are also maintained, and actually by the time you get out to this point where you have suppression of reactivation, basically, of that long-term RNA, those B cells are the most prominent cells that are present in the nervous system. So what are these cells doing? Next. Yeah, next. So now we're looking at these same, we're looking at the same brains of the same animals over the same period of time, but now we're looking at cells that are actually making antibody. So using an antibody ELISpot kind of assay, you can quantitate the number of antibody secreting cells that are present in the brain. And furthermore, you can know whether they're specific for Synbus virus or not. And in this case, we're only looking at the ones that are making antibody to Synbus virus. And we can also determine whether they're making IgM, IgA, or IgG. And what you find is that early on, all of the cells that have come into the brain during this first 8 days are making IgM. And only a small proportion of them are actually making IgM that's specific for Synbus virus. But then there's a steady increase in the number of IgG secreting cells and IgA secreting cells that are then maintained. So early on, it looks like IgM may be the most important antibody for suppressing infectious virus and clearing infectious virus from the brain. And that's a hypothesis that we're actively testing at the moment. Next. Next. So if you look then at the percent of cells, as I say, we can determine both how many cells are making antibody of any variety, but also how many cells are making antibody specific to Synbus virus, you find that, that the proportion of the cells that are coming in that are making antibodies specific for Synbus at the beginning is quite low, but steadily increases. So the B cells that are retained in the brain that are antibody-secreting cells are steadily enriched for cells that are actually making antibodies relevant to the infectious process. So really 100% of the IgG secreting cells are making antibody to Synbus virus. Furthermore, if you look in the brains just for the amount of antibody, not just the numbers of antibody secreting cells, but you just grind up the brains and say how much antibody is there to Synbus virus, you find that there's a lot of IgG, and that steadily increases really up through 3 or 4 months after infection, and then it's maintained. The amount of IgM decreases, not surprising, and IgA is intermediate. So there's a real enrichment of these Synbus-specific cells, and they're retained and maintained in the brain for a long time after clearance of infectious virus and after RNA has been decreased to quite low levels. Next. So we think that we put together the clearance of RNA viruses from neurons. You get the neurons infected. There's a period of control and clearance of the infectious virus that occurs that is dependent on both antibody and on interferon gamma. Also, I'll mention that type 1 interferon is also important, and all these animals do make type 1 interferon, but it doesn't clear. It only puts a cap on or controls virus replication. And then there's long-term control, where you have continued presence of B cells. I haven't talked about T cells, but well, I showed you in that one graph that there's still T cells, particularly CD4 T cells, that are maintained in the brain for long periods of time. So this long-term control then is also necessary. Next. And this just says in words pretty much what I've said, and that it's the antibody to the E2 glycoprotein, so a surface glycoprotein, and interferon gamma that mediate the clearance of infectious virus. The anti-E2 antibody restores sodium pump function and host protein synthesis. Interferon gamma decreases the viral RNA and protein synthesis, a JAK-STAT-dependent pathway. Clearance of infectious virus coincides with the entry of CD8 T cells and IgM-secreting B cells. And we're hypothesizing that IgM is more important than IgG. And if you remember the cross-linking experiments, where bivalent antibody was much more functional than monovalent antibody, maybe pentavalent antibody is even better. So that's one of the things we're looking at. And suppression of virus reactivation likely requires the long-term maintenance of these antibody-secreting cells in the brain. So next. So, but you might say, I mean, neurons are long-lived cells. There's a lot of reasons you don't want to kill them, that the immune system needs to figure out a way to be able to control infection in neurons without killing them, because they're not going to be replaceable. So maybe this is not particularly relevant to other sites, other viral infections. Next. So this is where measles comes in. And it was really based on some of these studies and really some chance observations in children in Zambia that we became interested in whether measles really got cleared the way we always thought it did. Next. So measles, a negative strand RNA virus, again an enveloped virus. The two most important proteins are, again, the surface glycoproteins with respect to the immune response to the virus. Next. Next. So it causes a rash disease in childhood, as most of you are familiar. And you may or may not be realize or remember or notice that it remains one of the most important causes of death in children under the age of 5. And of vaccine preventable diseases, it's very high on the list of viruses that and the list of viruses that cause death in children that could be prevented by vaccination. Next. So the complications of measles that cause it to be a lethal disease, most childhood rash diseases do not cause a lot of mortality, but measles does, particularly in developing countries. And what is it that actually causes those deaths? So these are the main complications. So the top three, diarrhea, otitis media, pneumonia, next, are other, basically due to other infections, next. And so these are this immunosuppression that occurs during measles, and most of the deaths are really due to pneumonia rather than measles itself. But there's a couple of other, encephalomyelitis is an autoimmune disease that we really don't understand well, and subacute sclerosing panencephalitis, next, is commonly, fondly known as SSPE, also is not a frequent cause of death, and this is a fatal disease. Next. But this is a disease that, it's a fatal disease of the nervous system that occurs 7 to 10 years after measles, and it's due to persistent measles virus infection and expanded measles virus infection. So children at the time they present with this disease, with cognitive dysfunction, et cetera, have measles virus throughout their nervous system. It's very widespread. And it's assumed that it entered during the acute phase of measles and then slowly replicated over this period of years before enough damage had been done that you saw neurologic disease. And so these are the big complications, and all of them they're not only important, but they're intrinsically interesting to try to understand how they result from measles virus infection. Next. So just to point out the immunosuppression, this is what happens to delayed hypersensitivity responses during measles by the rash. These are baseline levels. Rash, they're undetectable, and they come back slowly, so over a month or so. And those complications, the infectious complications, also occur over the next 1 to 2 months after measles, so it's not right at the same time necessarily. And this is another measure of immune suppression, lymphoproliferation, and it's suppressed for very long periods of time after apparent recovery from measles. Next. So the basic pathogenesis of measles, in brief, is that it's a respiratory infection, and then spreads, and a lot of replication of this virus in lymphoid tissue, and then, of course, in the skin to cause the rash. And then you start getting clearance of this virus, and this again is looking at infectious virus. And the types of cells, next. The types of cells that the virus is replicating in during this period of expansion of the virus are a number of different kinds of cells, but they're epithelial cells, endothelial cells, monocytes, macrophages, T cells, B cells. All of these are replaceable. So it's not like neurons where you really can't replace those cells. These cells you could replace. So if the immune system eliminated them, it seemed like that it wouldn't be a huge tragedy, that they could they could be repopulated. Next. Next. So this clearance process is coincident with the onset of the rash. And the rash, if you biopsy it, you find CD4 and CD8 T cells there. So there's an infiltration of immune cells into the sites of virus replication, which are undoubtedly occurring all over. We can just see it most easily in the skin. And you can measure a number of other parameters of immune response to measles that is occurring at the same time. So the question next is, do we really have clearance? So for a long time I drew this graph like this, and I did not have this little dashed line and question mark. But we began to wonder whether it was really cleared as efficiently as we thought by this immune response. Next. And so identifying the mechanisms of clearance we thought were important for a number of things. I pretty much mentioned these early complications that are due to immune suppression, the late complications that can be due to persistent infection, and then we also thought this may be a clue to lifelong protective immunity. Measles is sort of the poster child for developing lifelong immunity, and maybe having persistent virus around could contribute to that. So next. So our first clues that actually the virus did, at least the viral RNA, was present for a much longer period of time than we had previously thought, and certainly much longer than we could culture virus, because basically you can't recover the virus from any site after the rash has cleared. So similar to what we saw with the mouse brains, no infectious virus that can be recovered. However, if you look for viral RNA, and these studies were done in Zambian children, and we had originally done them because we thought some of the children that we were studying there were also infected with HIV, and so our hypothesis was maybe they won't clear measles as well as children who don't have HIV, and that was the main reason that we were even looking. And what we found was that 50% of the children, you could still easily detect measles RNA from some site, either blood, urine, nasopharyngeal washings, at a month. And actually, it was present in both HIV positive and HIV negative children. So then we thought, well, it must be gone by 3 months. And so we brought the children back at 3 months, and 35% were still positive then. So again, it suggested a very long process or period of clearance of the RNA after there was clearance of infectious virus. Next. So there was only so much you're going to be able to do with Zambian children and small amounts of blood and infrequent sampling. So we turned to our rhesus macaque model of measles, and they're the only, as I say, it's a human virus, so it's the only good animal model for this infection. And so they get a rash disease that's really very similar to measles in humans. And this red dashed line is, and the animals are infected intratracheally, so it's a respiratory infection. This is the viremia, so this is the amount of virus present in the blood. And this is recoverable virus that you can co-culture. This is how we titer it. But this black line is the RNA in those same peripheral blood mononuclear cells. So again, peak at the same time, a decrease, but then a very long period of time during which RNA was still present. So this provided us with a model that we're still very actively studying for trying to understand how this clearance occurs. And so I'll sort of update you on where we are. Next. So this clearance is occurring during the rash as shown on the previous sort of diagram. Next. So this is looking at 3 individual monkeys, and it's mainly just to show you that this is a period of time which you have infectious virus, and this is an RNA, and that the fact that there's, they all look pretty much the same. It's a very reproducible kind of process. Next. And furthermore, if you look at the respiratory secretions, you also have RNA present in respiratory secretions for many days after clearance of infectious virus from the blood. Next. And then we thought, well, we can't find it anymore after about day 60 or 70 in blood, but how about lymphoid tissue? I mean, we're only sampling a pretty small part of the compartment of immune cells. And so we looked at lymph nodes biopsies 71 days after infection. Next, next, next. So these are three different monkeys where, again, we have actually still large amounts of RNA that are present in the lymphoid tissue. So we don't know yet how long it takes to actually eliminate the viral RNA from all these tissues. Next. And I'll just point out that this is a period of time, this 2-month period is a period of time of immunosuppression. So we think that having the virus around might actually contribute to the suppressive properties and the long-term suppression that is known to occur in clinical measles. Next. So we have lots of questions. We're only going to try to answer 1 or 2 of them now. But there's lots of questions about the virus, where it is besides the lymph nodes, how long it's there, does it change, is the same virus there as it was at the beginning, etc. What's going on with the immune response, and that we're going to look a little bit about, especially with respect to the function, regulation of the immune response, induction of memory, and then eventually we have to prove that it matters. But next. So one experiment that was done quite a long time ago was to deplete monkeys of CD8 T cells and see what happened to their viremias. And so this is again just looking at infectious virus, not at viral RNA. These monkeys were studied long before we knew that viral RNA persisted for such a long period of time. And you can see that the viremias are higher in CD8 depleted animals than they are in control animals, and clearance therefore takes somewhat longer period of time. So we think there's pretty good data. CD8 T cells are important, so that's probably one component of virus clearance. Next. And then these studies then looked at these monkeys that were followed for longer periods of time, not depleted of anything, just normal monkeys, naive monkeys that were infected. And now the red line is the viral RNA that I showed you before, and now this is looking at T cell responses at basically interferon gamma ELISpot. So sort of a basic look at what's going on with T cell responses during this period of virus clearance and viral RNA clearance. And what's interesting is that as the T cell response comes up, the virus goes down. As the T cell response goes down, the virus goes up. And there's this sort of cat and mouse kind of pattern that we're seeing with the T cell responses and the virus levels, suggesting that regulation of the T cells or downregulation of the T cells may be an important component of allowing the virus to continue to circulate. Next. And a number of things are happening with the immune response as you, during this period of time. So the IgG responses are rapid. Next. But the maturation of these IgG responses, next, is very slow. So you get high titers within about 2 weeks. But at that same time, the avidity of the antibody is very low, and so the quality of this antibody is not great, but it steadily improves. So there's a lot of evidence that the immune response is maturing as time goes on. Next. Also, the T cell response. These circles are basically telling you how many different cytokines a T cell can make, and it looked at different times after infection. So these T cells can make 4 different cytokines. Most of these guys are only making 1 or 2. And so you also see a maturation in the T cell response that's occurring over time, well after infectious virus is cleared. Next. Next. And a look at the regulation, and we looked at the regulation in a couple of ways. First of all, you can look at the cytokines that are being produced, and interferon gamma is produced early. But late, after clearance of infectious virus, you can see regulatory cytokines being made, IL-10, IL-4, things that may suppress the Th1 type of response, so suggesting that this response is being regulated. Next. Next. Oh. So early on, you have a Th1 type of response, and we know we have activated CD8 T cells during that period of time. Then it shifts to a type 2 response later. Next. And that persists for actually a long period of time after virus has been cleared. Next. We also know that regulatory T cells are induced. We've done a lot of measurements of FOXP3, a transcriptional factor that's important for regulatory T cells. This shows another way of measuring it, which is CD4, CD25. There's an early peak that is not shown here, was not actually measured in these animals, but has been subsequently measured. But then there's a later peak. So regulatory mechanisms are being induced during this immune response and clearance process to measles. Next. So at that point, we were a little, I don't know, we weren't going to be able to figure all of this out, first of all, from these monkeys, and we had lots of measurements, but how do they fit together, and who's doing what, and what's important? So at that point, we currently are working with a couple of mathematical modelers at Princeton, Roger Kuyas and Brian Grenfell, to help us put together the different pieces and see what's important, see if we can determine what's important, and also maybe determine what the best questions are to ask next. It's, oops, no, sorry, not next, previous. Yeah, thanks, sorry. So we had 3 parameters. One is, well, 4 parameters really. One's a virus, and so that's the amount of viral RNA that's there, and that's going to keep replicating, we assume, unless the immune response controls it. So our immune response controls are, first of all, an antibody response, second of all, a T-cell response, and they have different patterns. And then third, a regulatory T-cell response, which will regulate this. And so those were the parameters that went into the model. Next. And those were all given, basically we're looking at the amount of virus, change in virus over time using viral RNA. And again, these same 3 parameters. Next. And so this just shows for 1 monkey, but it's true for 8 monkeys, that the spots are the actual RNA levels. The red line is if you only put in the T-cell response, which does not. And we're looking for a line that will actually predict what our data are. The green line is putting in the T-cells and the antibody. That's better. But if you put the but you don't see this hump that we do see in the viral RNA. But if you put in the regulatory T cells as well, you now start to be able to more closely mimic this. So this is an approach we're taking. We're refining it. We're there's a lot of different ways to refine it, and hoping that this will help us understand what the important parameters are. Next. So the conclusions are next. The clearance of measles is slow. It may occur at different rates from different tissues. I didn't talk about that, but I think that it's likely that it may be cleared from certain types of cells faster than others, and we have some preliminary data that suggests that's the case in the blood. And we don't know when it's complete. Next, we need to do longer term studies. The appearance of the RNA and the T cells is multiphasic, and it looks like it could be premature T-cell downregulation before the virus has really been controlled. If you shut off the T-cells too early. Next. And the maturation of a measles-specific immune response is gradual. Avidity maturation, T-cell maturation is occurring well after the infectious virus has been cleared. Next. And this mathematical modeling shows that both antibody as well as effector and regulatory T cells determine the level of viral RNA in the blood. Next. And these are the people that have been involved. I won't go through all their names because it goes back to Beth Levine did the original studies with skid mice and clearance. And Taliba Metcalf did the most recent studies on the B cell entry into the brain and T cell entry into the brain. And the measles studies have been long-term studies with 2, a postdoctoral fellow and a graduate student. Bill Moss is the epidemiologist we've worked with in Zambia, and Bob Adams is our primate veterinarian with whom we could do nothing, and I mentioned Roger and Brian at Princeton. Thank you. Do I get questions? Sure, I'm glad to take questions. If I can see anybody. Yeah.
Yes, a couple of questions. They're sort of related. So in the case of the CIMD virus, you saw IgA detected antibody-producing cells, IgA, in the brain, right?
Right.
Did you see Tregs associated with that? And that sort of ties in with the later question. When you see the Tregs in the measles, do you see IgA? I mean, you're tracing the.
Yeah, I haven't tried to. First of all, we haven't looked carefully for Tregs in the brains of these mice. T-cell, we focus more on the B cells and haven't really done a lot of analysis of the T cells that are coming in, either CD4 or CD8. And for the monkeys, we've done the Tregs, and we know they do make IgA, but we haven't looked at what that correlation might be. So what are you thinking, with the Th17 or?
Well, perhaps. I mean, just out of curiosity, since the TGF-beta is associated with the switching to IgA, and
right.
As a possible source of TGF-beta. I'm not saying it's the only one, but when you try to correlate in some cases the Treg response with
Right.
secretory IgA production.
Yeah.
Maybe find a correlation.
I don't know. It's an interesting question because IgA, I mean, first of all, we don't really usually expect to find it in the brain,
Right.
but actually when
Exactly.
people look for it, it's almost always there. So it is a characteristic of the antibody response to CNS infections that IgA gets produced locally. But I haven't really dug into why that might
Well,
be.
the fact that you have relatively low numbers and would go along with the way you may not need many Tregs to get in.
Right.
So that could also be of interest.
Yeah, good idea.
Thank
Yeah,
you.
Great.
So along the same lines, I was wondering when you did the addition of the antibodies with the skid mice with the same model, did you look at the microglial activity and maybe they were part of the reason why it was so protective once you added the antibodies?
Look at what kind of activity?
Microglial cells?
Oh, in those animals, there's a lot of microglial activation
Right.
that occurs
Right.
in these infected mice. I don't think that we ever look to see whether, when you do the passive transfer of the antibody, what effect that had
Right.
on that to microglia. It'd be an interesting question, but.
It's a lot of great data, and I probably should be asking a more sophisticated question, but my interest is with your measles work showing persistence of RNA and somehow possibly linking that to the persistence of immunity. Do you think that was based on in vivo studies? Do you think that may also be occurring with vaccination, intramuscular?
Yeah, well, we're doing a bunch of vaccine studies now, and you don't get nearly the replication of the vaccine virus that you, or the spread of the virus, particularly in the immune compartment and lymph nodes, et cetera, that you do with the wild type virus. But we're interested in that, and we're puzzled by that. So I do think that the vaccine may persist, but vaccine virus, RNA may persist, but it's certainly just overall is at much lower levels than, and there's no clinically significant immunosuppression that occurs with the vaccine, with vaccination. So we don't really know. As I say, we're just in the middle of really trying to initiate some of those studies.
I think that's interesting because it looks more and more like in this era where circulating pathogens have been nearly eliminated from many of the childhood diseases. We're seeing more and more waning immunity and the need for boosting, where with measles, it almost surgically searches out those that are not vaccinated for an imported case and infects them. So I wonder if that's just a property specific to this pathogen. It'd be something interesting to look at.
Yeah, you do get waning immunity after the vaccine, but it's really you get pretty durable immunity after measles vaccination, but there's a subset of people where it does wane. Although, interestingly, if you revaccinate those people, it goes up and then it goes right back down to where the level was before, so.
I'm curious to know, when you see the persistence RNA, what component of the virus is that? And is it a structural? Is it a functional? Does it matter? Do we know?
Yeah. So good question. And all I can say is we don't know as much as we'd like to. We would really like to do whole genome sequencing across these different time points, but got to get some money to do that. But yeah, or a good collaborator with money. But we've PCR'd from different areas of the genome. We know that. So if we go back to the SynBus model, we know that both structural and non-structural RNAs for both structural and non-structural proteins are there. We also know in the skid mice that if you just give them one dose of antibody and then follow them, the virus will begin replicating again about 3 or 4 months later. So what's there in those mice, at least, is able to resume replication. So at least a portion of it is whole genome. In measles, we have pretty much the same kind of data. We've sequenced, or we've PCR'd from at least 2 areas of the genome, and we find both. But we haven't distinguished whether it's plus or minus strands, so whether it's genome or message, which I think is an important question still. And we are very interested in knowing whether it's escaping, from immune control as a part of this. But again, we don't know. And we don't have any way for those for those animals to know whether they would, whether the virus could resume replication or not. But I'm assuming that proteins are being made, and at least message is being made whether the virus. These cells turn over pretty fast. Epithelial cells, monocytes, T cells. I mean, I would have thought, although it's mostly in a memory T cell population gets infected most, but I guess I just would have thought that that must indicate ongoing infection, but we don't have any good proof of that.
Well, thank you, Diane, for a wonderful lecture. And for those of you who can join us, please join us in the library for the reception.
Very nice. Oh, thank you.
Thank you.